so our last concept for chapter 6 is the cytoskeleton and extracellular matrix. So I want to show you guys this video of what's happening inside a white blood cell as it starts to move around. All right, let's see if we can get this to go. All right, so we're zoomed in on the surface of the white blood cell that's interacting with the blood vessel wall. Now we're looking at the phospholipid bilayer, and hormones are activating proteins on the surface of the cell that activate proteins inside the cell. Now this is the cytoplasmic side. These are actin microfilaments that give the cell shape. The cytoskeleton is very dynamic. It changes, it grows, it shrinks, and so it gives the cell shape, but it's not permanently in place. These actin microfilaments form quaternary structures. They can build together and grow. They can also be cut apart and shrink, so the cell can change shape. These are microtubules. Now nothing travels in the microtubules. They can also grow and shrink. They're dynamic. And they're the railroad tracks for motor proteins that are dragging vesicles of the endomembrane system around. The proteins really look like this. They look like little guys of 2 or 3 feet. The microtubules grow out of the centrosome, made of 2 centrioles near the nucleus. Now we're zoomed in on the nuclear envelope. Here are the nuclear pores, and RNAs are leaving the nucleus. They actually form little circles with translation initiation factors, and then the 2 subunits of the ribosome assemble and read the mRNA to make a protein. This protein is being trafficked to the mitochondria. Other ribosomes are going to sit on the surface of the rough ER and push their proteins into the lumen of the rough ER. Then vesicles are going to pinch off of the ER. They don't just float through the cytoplasm though. Those vesicles are brought from the ER to the Golgi by motor proteins walking along microtubules. So those proteins are getting their sugars modified in the Golgi, and then those vesicles are transported to the plasma membrane where they're either released and secreted or make up proteins that are embedded in the plasma membrane. And these allow for cells to interact with one another. For example, when they receive a signal from the blood vessel, which activates these proteins, which make all those changes inside of the cell, which allows this white blood cell to slow down, stick to the blood vessel wall, and then squeeze between the cells of the blood vessel and leave the bloodstream to go fight an infection somewhere else. I love that video because it does a good job of illustrating the cytoskeleton. So the cytoskeleton is a network of protein fibers that give the cell shape and support, but also allow cells to move around. And so there's 3 different cytoskeletal components I want you guys to know about: the actin microfilaments, the microtubules, and the intermediate filaments. And we're going to talk about what each of them do for both the structure and function of the cell. So the cytoskeleton helps the cell move along with the help of motor proteins. So motor proteins use energy released from hydrolysis of ATP to change their shape, which allows them to literally look like they're walking around within our cell. So we're going to see how motor proteins with cytoskeletal filaments help the cell move. We're going to talk about each of these. We're going to start with the microtubules. Microtubules, they're really small, but they're actually the largest of the cytoskeletal components. So the microtubules are made out of a protein called tubulin, and there's 2 different isomers of tubulin that assemble into these tubules. Now, there's nothing that goes through the tubules. The microtubules make these railroad tracks for motor proteins to move on the inside of the cell. So especially microtubules and actin microfilaments are dynamic. They grow, they shrink. And so the side that grows and shrinks is called the plus end, and the side where they're anchored is called the minus end. Microtubules are anchored to the centrosome. The centrosome is made of 2 centrioles, which are at 90 degrees from one another. And it's usually located close to the nucleus, and then the microtubules radiate out from here all around the cell. One way that microtubules help cells move is with the help of their motor proteins. So motor proteins walk along the microtubules and transport things within the cell. We saw that in the video. There are different motor proteins that work with microtubules. So kinesin actually walks from the centrosome out to the edge of the cell, and then dynein actually walks from the edge of the cell back toward the centrosome. So one function of microtubules is they do transport of vesicles within the cell. Another function of microtubules is they help coordinate the movements involved in cell division. So microtubules, along with their motor proteins, anchor to the chromosomes and pull them apart when the cell divides. Microtubules also overlap with each other, and different motor proteins push each side of the cell further apart. And then other microtubules anchor to the cytoplasm and pull the centrosomes out to the edge. And so these coordinated movements help the cell divide. One more function of movement with microtubules is supporting cilia and flagella. So some cilia line our lungs and move fluid past the cells. Flagella have the same underlying structure that allows cells to swim through fluid, like in sperm. But there are also cilia that play a structural role. So in our rods and cones in our eyes, they're supported by cilia. There are cilia in our ear cells that perceive sound. And there are primary cilia in most of our cells, which are involved in left-right body pattern formation in embryogenesis. Whether it's a cilia or flagella, the underlying structure is the same. There are pairs of microtubules with motor proteins in between them. And it's the movement of these motor proteins, which are anchored on one side, that slide one set of microtubules past the other. And that allows the cilia and flagella to wiggle back and forth to either move fluid past the cell or propel the cell through fluid. Microtubules help the cell move by transporting things within the cell, making railroad tracks for motor proteins to walk along. They help with the movements of cell during cell division. They support cilia and flagella that help cells move, and also they just support the overall structure of the cell. So the next cytoskeletal component we're going to talk about are the actin microfilaments. The actin microfilaments are the thinnest cytoskeletal component. Okay, so microfilaments and microtubules sound very similar, so I tried to specifically say actin microfilaments. Actin is the name of the protein that assembles to make the fibers, which are called microfilaments. So actin microfilaments are also dynamic. They grow, they shrink, they move. They support the overall structure of the cell, but they also help it move. So just one example of the structure. Actin microfilaments push the plasma membrane out in these fine little finger-like projections called microvilli. And it's these microvilli that increase the surface area of the cell to allow for more absorption in tissues like our digestive epithelium. So actin also helps our cells move with motor proteins. So we're going to watch some videos of cells moving with actin and their motor proteins. So in this video, we have a white blood cell that's chasing down a bacteria. The bacteria is making a waste product called cyclic AMP, and wherever that concentration is highest, the actin microfilaments are going to push the plasma membrane forward and cause the cell to ooze in that direction. And eventually this white blood cell is going to phagocytose that bacteria and eat it up. The motion that you just saw is called amoeboid motion. What was happening is actin microfilaments were pushing the plasma membrane out. And the places where the plasma membrane gets extended is called a pseudopod, a sort of foot. So the structure supported by actin microfilaments is called the pseudopod, and the movement is called amoeboid motion. So actin can help cells move, and with its motor protein, it can make movements as well. So actin, with its motor protein myosin, can do contraction. So this is what moves our muscles macroscopically. The actin filaments remain in place. The myosin motor protein is anchored, and so when these little feet flex, they pull each side of the sarcomere together and shorten the muscle. So actin and myosin help our muscles move, but a similar structure, a band of actin and myosin, actually pinch cells apart. So this can move our cells, move our muscles, but it can also help in cell division, forming the contractile ring. That little spot where the two cells are getting pinched apart is called the cleavage furrow. And this helps cells divide. And then, of course, actin also supports the overall structure of the cell. The last cytoskeletal component are called the intermediate filaments. They're called the intermediate filaments because they're intermediate in size. They're the middle-sized ones. They're actually probably the most boring cytoskeletal component because once they get laid down, they're really locked in place. So they're the most skeletal. They move the least. One structure that they specifically support is the nuclear envelope. They make this network of protein called the nuclear envelope that holds the nucleus shape up. So the last concept for chapter 6 is the extracellular matrix. The extracellular matrix is a structure found outside of cells. So for plants, the extracellular matrix is the cell wall, which is made of cellulose. And the cell wall is perforated by these holes called plasmodesmata. Plasmodesmata allow for the exchange of fluid, sugars, RNAs, ribosomes in between plant cells. In animals, the extracellular matrix is a network of highly glycosylated proteins that anchor cells to one another and to this spongy protein carbohydrate matrix. Cells get anchored to each other through junctions. And so I do want you to know these different junctions and how they're anchored to the cytoplasm. Tight junctions make watertight connections between cells, so no fluid can squeeze between the cells of our gut epithelium. All the nutrients and water have to go through the cells. Desmosomes are point connections. They're used in tissues that are under a high amount of mechanical stress. So in skin and heart, these desmosomes anchor cells to each other. And on the inside of the cell, the desmosomes are anchored to the intermediate filaments. And then finally, gap junctions are holes between cells that allow for the flow of fluids, sugars, hormones. Gap junctions are protein-lined holes between animal cells. This is similar to the plasmodesmata in plant cells, but those plasmodesmata are called plasmodesmata because they're lined with plasma membrane, whereas gap junctions are lined with protein. Gap junctions are particularly important in our liver cells that allow for signaling on glycogen levels and also in certain neuronal circuits in our brain. So for the different junctions, you should know that tight junctions are watertight and they're anchored to the inside of the cell through actin microfilaments. Desmosomes are used to anchor cells together under high mechanical stress, and they're anchored to intermediate filaments on the inside. And gap junctions are holes that anchor animal cells together that allow for the flow of fluids, hormones, sugars, and other small molecules.